Hello. So thanks to the organizer for having me here. Yes, it is a long way. It's 24 hours in the flying car. So what we're doing, yeah, if you live down there, you do it regularly, so you get used to it. Anyway, so what we do in our lab, what we do is basically, we normally do technical development, method development, and specifically around cryo-EM. Effectively what we do is that we take microscopes and we break them. So initially I thought I'm going to talk about single particle, but that's so last decade that I'm going to focus more onto the cellular tomography. And I mean, a lot has been said already by Julian, by Wolfgang yesterday about how lamellas are prepared and how basically what kind of information you can get. So what I want to share with you is basically our latest finding in the latest focus ion beam technologies. So, and specifically on the plasma FIB. So I'm going to show a little bit on plastic, because still most of the people actually are doing plastic. Not everybody does cryo, and then basically we're gonna move a little bit toward the in situ structural biology. So if anybody hasn't seen a dual beam before, this is how it looks like. At least this is one of the ones that we had at Monash. So you have an electron column which is top down, and then anywhere between 38 and 90 degrees you can have a focus ion beam. The focus ion beam, basically what it does is you have a flux of ions which are accelerated and they collide onto a sample, and basically upon collision what you have is typically you're sputtering away the atoms which are forming the sample. So, and with the atoms you're going to have generation of secondary electrons as well as secondary ions. These can be used for imaging, so you can see what you're sputtering, as well as another thing that will happen, which is pretty important, especially in the context of material science, is that the primary ions from the beam will be implanted into the material. So, what are the typical uses? Typical uses are FIB-SEM tomography. So you're gonna essentially what you will do is that you will image with the SEM, then you will shave away the surface, then you will image again, and so on and so on. And this will basically provide you with a volume of all the electron densities in your sample. Another very common thing is to do is basically prepare samples for TEM. So you have a sample which is too thick for transmission electron microscopy. What you will do is that you will thin it down, and then it will be compatible with TEM. In this case, just to highlight what the basically the implantation effect is, this is diamond, and if you cut it with gallium, which is a metal, what happens is it will implant, and you will see this junk. So you don't see a clean lattice, you see a lot of defects basically. That's due to implantation. If you use a different source, this doesn't happen. Other things that you can do, this is more of gallery things that we do all the time in the lab, is for example you can use microfluidics. So you can do microfluidics, you can directly mill materials and you can make your channels, you can do microelectronics, you can do mask writing in case you are into microfabrication. You can do grayscale lithography. This is basically one of the major focuses of my lab lately for a reason that I still have to understand. So we're doing all sorts of fancy lenses. So these are hybrid refractive optics. You can see like multiple spots coming out. You can make photonic vortices, which sounds like a very cool weapon that teenagers would, actually kids more, would play with. And then you can make butterfly lenses, or we don't really know how to call them yet, so they make these spots. You can do grayscale lithography, and so here just I'll digress a little bit from the main topic. So you could do Fresnel zone plates, and in theory you should. So Fresnel zone plates are used in X-ray for focusing the beams. This is just a short parenthesis. And so basically, normally what you have is basically you have a pattern which is repeated. The pattern is normally binary, so you have areas which absorb and areas which basically don't. And the finer, so basically the zones will get finer and finer and finer. And basically the higher is the frequency of the zones, the higher will be the diffracting angle, which will basically define the numerical aperture of the lens. Okay, these lenses here are the ones that are used, for example, in X-ray tomography. If you need a lens, the objective lens will be a Fresnel zone plate. They are very inefficient, so they are 10 to 20% efficient normally. So it means that most of the photons will actually not be focused. So there was in theory, we were supposed to get 100% efficiency, and this basically, if you do essentially Kinoform lenses, which basically means that they have a parabolic profile which goes across the zone, so from the 0 to the 100. That in theory is the case. So we thought, oh, we can do grayscale lithography, cool, let's do that. So we did it, and this was the output. So the Kinoform was bad. The other one was better, still bad. And we started wondering why that's the case. We spent a lot of time doing this, we did a lot of simulations, and basically we killed the field. So you should not be doing Kinoform lenses, because essentially for a very long time the math that was used was wrong, and they did not take into account absorption, unfortunately. And basically, if you think about it, so if you do if you basically design your zone plate to induce a 2-pi shift in the phases, and you don't consider absorption, you will get 100% efficiency. But the truth is that absorption kicks in, because photons will get absorbed, so you will get nothing more than 30%. So it's not much better than what you normally get already with binary zone plates. So we did a lot of measurements, and basically at 6 kV in this case, because that's where our detectors was optimized, we could get basically the theory and the measurement to match. So we've run more and more simulations, and we figured out that basically, in theory, you could get to 70%, but only for absolutely impractical heights. So, for example, 4 micron height. So if you want to achieve a zone width of 50 nanometers, this is just, it's not going to stand. And it will only work in areas around like 10 kV or so. We've tested it, we've simulated many materials. So essentially, these are not, it's just like if any of you was into zone plates, don't do this. Anyway, other things, now let's move to something which is more of interest for this community. So, what I said, I'm going to be talking about plasma FIBs, and first I need to explain how focused ion beams work and what's the difference between plasma FIBs and more conventional liquid metal ion sources such as gallium FIBs. So this is a liquid metal ion source. So this is the schematic. So you're going to have a tungsten filament which will be coated with a liquid metal, could be gallium, could be gold, doesn't matter. You will apply a very strong electrical field, and then basically what will happen is that ions will be extracted essentially from the tip of the filament, and then it will be accelerated and focused onto your sample. So what's very important here is that what you can do is have very high resolution. So your tip will define the source size, which basically means that you'll be able to have a spot size which is down to a couple of nanometers. But your maximum current is going to be very limited, because your source is very small. So there will be a limit to how many ions basically you can extract at one point. And with the increase of the current, what will happen is that you will lose coherence, so you will lose focusing efficiency. Another thing is what you can do if you need to have a lot of current is you can use plasmas. So plasmas are not very hot. They're just highly energetic. First of all, what happens is basically you have this source, which is a leaky vacuum chamber. You leak into the chamber whatever gas that you like. You apply a radio frequency, and that will basically will turn your molecules here, your atoms, into a plasma. So they will be highly energized, and then you will use an electrical field again to extract basically your ions. The main difference here is that what comes out from this orifice, which is your source, is a collimated beam, and the size of of your source will be limited by how small can this orifice be. So main advantage is you can get currents which are in the order of 2.5 microamps, not nanoamps, like in this in the liquid metal ion source. So just these are measurements that we've taken between different instruments, and just the take-home message is, if you need to work anywhere between 1 picoamp and 10 nanoamps, buy a liquid metal ion source. If you want to work beyond 10 nanoamps, buy a plasma field. And just because we cannot have anything normal in the lab, we have so we can switch the ion source in this guy across all these potential sources within 5 minutes. So we just switch the gas and just light up a new plasma. So we thought, okay, we got all these plasmas, let's do something cool with it, and let's test how they behave with, in relation to biological samples. So we started with looking at plastic, because 90% of people in EM do plastic, they don't do cryo. And so we basically took resin, epoxy, 2 epoxy resins, 2 acrylic resins, and we started milling them. We used xenon. Why did we use xenon? Because it's the heaviest plasma you can light up, and so it's very efficient in sputtering, and it's the only commercial gas. So everybody that has a plasma FIB will have a xenon FIB. Okay. So what we found out is that epoxy behave okay, acrylic don't. So the xenon doesn't really work on acrylic resin. Okay, this was it. We thought, okay, let's go for something which is a bit lighter. It's normally used for polishing. Material scientists use argon all the time for this. Let's see how it works. It works kind of a bit better. It's kind of HN20. It's usable, but it does this funny thing on the surface. So it's not impressive. Let's put it that way. And it's freakingly slow. Then we went to oxygen because we thought it's carbon-based. Let's do some etching. Works beautifully throughout. Everything is just fantastic. No matter what current we used and no matter what material we used, the cut phase was always curtain-free. This is really promising. Then we compared it to gallium, because gallium is the benchmark, because everybody's using gallium. And we found that essentially gallium behaves very similar to xenon. LR white can be used. Nobody does it, but you could do FIB-SEM tomography on LR white, and HN20. Originally we thought we can't. Later we figured out that if you lower enough the current, you could still use it. So, but one thing that we did notice when doing some measurements, and this is basically, so this is an example, same sample across, basically, so it's mouse brain embedded in implant, stained with osmium. So what we found is that there is very little contrast if you're using a liquid metal ion source compared if you're not. So the contrast was improved if we were not using the liquid metal ion source to measure. And we tried to quantify this. We figured out that basically by doing taking measurements of the local variance, which is basically tells you how much noise you have locally, what we could see is that the distribution of the local variance in images that were acquired after milling with a liquid metal ion source, it was like there was more noise essentially. And the reason is because of the implantation, as this phenomenon was explained at the beginning. So what happens is that you have gallium, right, which is heavy. It's a metal, it's heavy, and but it's not as heavy as the resin, which is made mostly by carbon and nitrogen. And it's also not as heavy as the osmium, so it's just in between. And implantation is a phenomenon which just happens randomly. So basically what happens is that you are lowering the backscattering probability in areas which are osmicated, and you are increasing the backscattering probability in the background. So basically you're just turning your nice image, crispy staining, into a noisy phase. So this is actually really interesting, because basically if somebody's interested in taking very large volumes, you should bet, and you're not interested in resolution, you shouldn't be using a FIB. You should be using a slicer, roughly, or a plasma eventually if you got one, because you will increase your throughput by 30% roughly, just because you can image much faster and have the same level of noise. Now, since most people here seem to be interested in cryo, we look a little bit at that as well. So this is, I'm going to go very quickly. So these are cells which have been plant frozen. These are yeast. They are not transparent. So if you need to do TEM, you're going to have to do something. So you have two choices. One is cryosectioning. Well, it's a beautiful technique, but nobody really wants to do that because it's very hard to get samples of good quality, and your thickness in the section is limited to 50 to 70 nanometers. So nowadays, basically, everybody's doing focused ion beam. What we know in terms of focused ion beam milling is that the throughput is not quite high. You're making 10 lamellas, 20 lamellas a day in some cases, but it's very low. So we thought, okay, let's, you know, we were so fast in cutting all this resin, and it was cutting so nicely with oxygen, for example. We could actually try and see what happens. So we thought, okay, let's do something massive. So we take a Drosophila embryo, we high pressure freeze it. In this case, we just exposed it by sort of freeze fracturing so that we could take pretty pictures. But let's see how fast we can obliterate part of it. You know, so we can measure what the removal rate is. So we used gallium, and you see, after 2 hours, 65 nanoamps, which is the maximum current I can extract from a gallium FIB. This is what you get, you know, all these pillars, and it just doesn't go away. So this actually happens after pretty much 20 minutes, and then nothing changes. Then we use xenon. Xenon is a bit better. Like, you know, in 7 minutes we can cut this little thing, 16 nanoamps, but that's about it. And the surface is really rough. It's not very usable. And the reason here is because what we are doing is that we are curing the so there is a lot of redeposition, and we are curing the redeposited material. So it's getting harder and harder and harder. On top of that, it's charging. So the whole thing here, which is not conductive, becomes positively charged. And I'm shooting a positive beam. And everybody knows that you can't quite reach very easily if you got two positive charges. I mean, they won't collide. So what we thought, okay, let's do oxygen. You know, it works so well, and it does work actually. I mean, in 2 minutes at 38 nanoamps, because that's the current that I can extract in that range. We actually can cut all this. You know, we can make a lamella throughout a whole embryo. It's not a nice lamella. It's not a lamella you would use for TEM, but I can cut through. So that's actually nice. I thought, okay, that's cool. We can do lamellas with oxygen. No, because physics is a bitch. And the reason is that, you know, you have an ion. It comes in. The lighter it is, the more it struggles into the material, and therefore what happens is that you're damaging. So the take-home message is, if you've got a very light atom, you will damage a lot on the side, and if it's a high energy, it will damage even more. So basically this is what's happening, you know, and this happens also with gallium, and this happens with everything. It's just that the heavier, the lower the damage. So what happened here is, you know, you have 300 nanometer lamella, and you start shading the, you know, both sides, and you're going to have a damaged layer. Well, this damage layer will depend on which atom you're using, which energy you're using. So if you use oxygen at 30 kilovolts, what happens is that you are actually damaging 140 nanometers per side. The lamella is 300. There's not much left. So we started looking, you know, xenon is very heavy, shouldn't damage. It curtains like hell. It's not usable. Yeah, so it's in success. That's it. Let's look oxygen. You know, 30 kV as expected doesn't work. 16 kV and 10 kV start working. You know, we actually get lamellas which are usable. The problem is that at 10 kV with oxygen we get almost no generation of secondary electrons, so we don't see our lamella when you cut it. So basically what's happening is that we can cut a lamella in 7 minutes. We are super quick, and then we take an image with the SEM and there is no lamella. Then we do another one, super quick. Bam, there is lamella. Yes, move to the next. Bam, no lamella, and so on and so on. Not very efficient. So now basically it's really good for fast removal, so we're thinking that it might make sense to use oxygen in lift-out, you know, for preparing for lift-out, so basically what Julia showed yesterday. Now, this is a cell, right, for biology. You know, you've got compartments, you've got proteins and molecules which interact and do things, but we're doing, you know, we're doing TEM, so this is a cell. So you need to look for volume. And what we will do normally is the following. We will take anything that's striping, we'll say this is volume. And then we will average it and we'll get a template bias, and we'll be happy with it. The truth is that if you have, you know, if wall is fluorescent, then you can take all the wallies, and that you know they are wallies, and then you know, they are all in different conformations, and you average them, and you can reconstruct wally, and you can reconstruct the variable domains. But so that means that we need to do some correlative microscopy. Essentially, we need to use you know, the information we get from fluorescence and translate that into our TEM analysis. So and this is how the workflow looks like normally. Okay, so the ideal workflow at least. So you go light microscopy, you freeze, then you do cryo-light microscopy, then you're going to be doing cryo-FIB, and since you're milling a 200 nanometer lamella out of cells which is 20 microns thick, what are the odds that my region of interest is still there? Well, not very high, to be honest. So you will have to do cryo-light microscopy again on the lamella, and then you'll have to do cryo-TEM. This means 4 cryo transfers, which basically means that at every transfer you will accumulate some junk, which will lower the probability of success in the end. So what we thought of, well, there are at least 2 transfers that can be removed, because the cryo-FIB is a big thing. Right? It's a big chamber, and it's mostly empty, especially because it's been designed for material scientists that have, Christmas tree looking like instruments with, with 500 detectors and everything around. Biologists don't have that. Most of the chamber is empty. So we thought, okay, let's stick in another microscope, and we've got 2 limbs, we can have a third. So that was version 1. This is version 2. It's a bit more professional, and it doesn't have an open beam shining. And basically that's how it looks like. So we plug in a fiber in here. We have our beam path. It's a standard wide field microscope essentially. We have a feed through for the light. The light comes in here. So this blue arrow here, and it goes to an objective which is positioned just next to the feed focused iron. And basically now we are in vacuum, so you can just translate from one to the other. This is basically how we control it. I mean, we have the microscope PC. We talk to the microscope PC through a Python API, and then we have our control, let's say control software, which basically allows us to take images with the light microscope, take images with the SEM. We save everything, so it already makes sort of a data structure so that you don't get lost into the data. And this is pretty much how you would do very quickly. You'll turn on, you say take image, it stores it here, then you say move from optical to FIB, and then we'll go to the FIB. You can see the movement down here. Then you can decide, okay, I would like to take an SEM, grab frame. This is what happens in the background basically. It takes a snapshot, it shows it here, and it saves it as well. Then you can say take a FIB, for example, image. It will do the same thing for the FIB. You can see it's coming there. And then, once you got your light microscopy image, another thing, you can move back to the light microscope potentially, and you could take multiple channel images in case your sample has multiple channels. So we can control up to 4 channels at the moment. Click a button, that's what happens. And you can say, okay, I would like to correlate now the last 2 images that I acquired. So you just click on the button, and that will give you this little control. So you can start, select. You can select your markers or areas that you can recognize being present in both, and then you click OK, and it will spit a correlated image in a second. Yeah, there you go. It will spit a correlated image, and then you can just use that correlated image to go and mill where you want. So how precise is this translation? So we went, from light microscopy to EM and back and forth and back and forth. And we put some fiducials, and we basically started, by picking bits in both modalities. We measured it, and we are confident that normally 90% of the cases on our stage we are within 400 nanometers away from where we expect to be, which is way more than enough to actually mill within a cell. Then you're still going to have to check your lamella anyway. This is fiducial free. You just take the 2 images and you overlay them. And if you want to get a better resolution, you need fiducials, and you click and you do the correlation with a better precision. So on biological samples, I mean, this is what could happen. For example, this is C. elegans, L1 larvas, which is pretty much the biggest thing you can plunge freeze, because it's full of fat and it freezes very well. And these are neurons, dopaminergic neurons, which are expressing RFP. What we do is that we decided to go and cut a lamella in this area. So that's how it is. We cut a lamella through the worm, and you can look. At each stage we could check whether our cell was still in the lamella or not. And basically we can go to the end of it, and we have a lamella, and we still have some fluorescence in there. This was a thick lamella. It was meant for X-ray. Didn't ever make it to the synchrotron because it cracked and died on the way. But it actually works. Another thing, I mean, for example, if you want to do cryo-liftout, which is what Julia was showing, I mean, it's really important that you're actually lifting out what you really want to lift out, because the whole process takes forever. So here is a Drosophila brain, which was also frozen, and what we did, we have some interneurons which are expressing GFP. We start milling and we mill, top and bottom, and we make lamellas which are ready for lift out. And we're sure that we're making them where we want them, on the spot. And we're sure that they're on the spot because you can see the spot in the lamella. And this is really handy. Then one thing that we were asked for when we submitted the paper was, can you actually see fluorescence out of the lamella, which is 200 nanometers? And the answer is yes. So this is a lamella, which has the proper thickness for TEM, and this is the fluorescence. It is limited. I mean, it's only 200 nanometers. Everything else is emitting from the whole cell, and the optical plane, because it's a wide field microscope, is about 1.5 microns in this case. So it's actually, we're going to have a difference in intensity, but it's still there, and we can take an image on the lamella, we can place it where we want, and then we can go and take a tomogram where we wanted to take the tomogram. So we wanted to be on the side of the nucleus, which is glowing GFP, and that's where we are, on the side of the nucleus. You can see a vacuole here, you can see ribosomes in the cytoplasm. So this is actually simplifying our life dramatically, and we don't have contamination any longer due to the transfers. So now I would like to conclude because I think the time is running out, and I would like just to basically summarize with, oxygen plasma seems to be the best, the most compatible beam in case you're doing resin embedded, if you're working on resin embedded samples. So essentially, my take-home message is like, if you're doing this and you need 5 nanometers resolution and you're doing very large volumes, go for it. It's fantastic. If you're doing cryo, it's a bit more complicated. Anyway, so gallium implantation does have an effect on contrast, and essentially by not using metallic ion beam, you can increase your imaging time. So you decrease your imaging time by 30% roughly. The oxygen plasma is the most efficient beam also when it comes to milling frozen hydrated sample, but it is problematic because it does damage a lot. So it only works for thick lamellas as of today. So it's probably something that will be very valuable for lift outs. And then the integrated light microscope is just working. So if you need one, just let me know. It can fit on pretty much any dual beam. And this is, thank you for your attention, and thank my team who basically did most of the work.